Today I'm going to give you hopefully a glimpse of where medicine and biology might be going forward, afforded by a powerful new set of technologies that underscore a new field of genomics. These technologies, this discipline is inevitably going to impact society. It already is beginning to do so. And we hope that it's always going to be for the better. But like in any technologies, there are some dangers which as citizens in a democratic society you should be fully aware of and be ready to govern. Now, what I'm showing you here is the substrate for all we're talking about. To your right is an X-ray crystallograph of DNA. In fact, that was the copy of the crystallograph that Rosalind Franklin produced that was used by Watson and Crick to do their monumental discovery of a model for how DNA actually looks like, the double helix. To the left is actually a crystal structure of that DNA, and this is also to describe how science is not only beautiful in concept, but also beautiful in figures. This, of course, is a structure of DNA. Now, many of you know already what genetics is all about. It's about your genes that you pass on to your children, that you receive from your parents. And the science of genetics is a study of genes and their functions, individual genes primarily and their functions. Genomics is just genetics on steroids. It studies all genes and how they function together. And the questions we ask as genomic scientists is to discover all possible genes involved in a cellular process or disease. Why genetics is front and center in all of biology, all of medicine, is that we're talking in biology about living forms. And there's only a few first principles of what life is. First of all living creatures replicate, mate, or divide. There is no living creature that does not do this form. The second, all living creatures evolve to become more robust. It's a basis of evolution, but this replication and this evolution, it's all based on some kind of nucleic acid template. Whether it's DNA or RNA, it guides all living processes, and there's a sanctity of the genetic code, which is ATCG, grouped together in groups of 3, most commonly called codons, and these combinatorics allow us to string near infinite possibilities in life. What do we mean by near infinite? It's the combinatorics of DNA and the proteins and the RNA that actually gives us remarkable functionality. I paired these up because they are actually related creatures. You have the elephant and a manatee that on a genetic basis are actually related, but one lives primarily in water, solely in water, and the other lives primarily in land. You have fish that fly and fish that live 1,000 meters underwater. And in bacteria, you have some very unique creatures called extremophiles. This Thermophilus aquaticus lives in boiling water. And the Deinococcus here actually was found in radioactive waste at rads that would kill any other living creature. Now, imagine this, that from the combination of 4 bases, you can generate creatures that are microscopic to macroscopic, that live in water and live in air, that live in boiling water and live in radioactive waste. This is really absolutely remarkable, and it gives you an indication of the possibilities once we harness the power and understand the code of life, which is in DNA and in RNA. I'd like to make a few declarations in my talk. First of all, I believe we're entering one of the most profound periods of advancement in biology and medicine, bar none, and one that will transform health, medicine, and equally importantly, is human sustainability. That is how we as a species will survive in the next 1,000 years. And it's all driven by advances in genetics and in genomics. The basis of this is your genome in medicine. It's 3 billion bases, organized in about 28,000 genes, with only about 1.5% of the genome that encodes genes. The rest are in a regulatory cloud. Used to be called junk DNA. We now know that majority of this is not junk, but how that works is still a mystery. We're organized into 46 pairs of chromosomes, 46 chromosomes in pairs. And all this information, all the genomics that we have is predicated on the generation of new and powerful technologies. First of all, new generation of ultra-fast sequencing technologies that allow us to massively generate data. The second is enabling computational advances that parallel the massiveness of the data generation, the analytical firepower that actually you're carrying in your cell phone and in your laptop. And finally, the effector arm of genomics is genetic engineering to create models of disease and, in some cases for medicine, the surrogate of disease. Just to give you an example of some of this technology, this is actually a histogram looking at the different sequences with each color representing a nucleotide, and the string where they are sequenced are arrayed to left to right. So you have a green here that is C, or blue that's C, and then you have 3 reds which are T's, and then double A and so forth. Originally, this was done on thin little capillaries using optics to actually separate each of those nucleotides. But if you take that and actually crunch them all together in a itty-bitty cell of 1 centimeter in size, this is what you actually see here, with every single dot that is actually a miniaturization of the capillary that used to be. And that allows for synthesis of 100 bases in situ. That is, in a 2-dimensional space, all these little dots will allow you to sequence 100 base pairs independent of one another. Now, in order for you to put this together, it's exactly like a very, very bad jigsaw puzzle. But computationally, we're able to do that. And it's only through computation that we can actually exploit the genome. Now, why is this so remarkable in biology? It's this graph here. This is actually the kilobases that can be sequenced per day per machine. A kilobase is 1,000 base pairs. Okay? Remember, DNA is sort of a linear molecule, ATCGs, and we're reading them, let's say, from left to right. But in 1980, when I first started out using manual gel slabs, it started here, but now, and projected in the near future, we have 7 orders of magnitude of increase in throughput. 7 orders of magnitude, that is 7 zeros. That is really unbelievable. In the history of biology, we have never seen this kind of advance in such a short period of time. Equally important is this reduction in cost of the human genome, this reduction of taking a genome that cost $300 million down to 3,000. At the beginning of the sequence of the human genome, the sequencing of the human genome that was completed in 2001 cost us, the taxpayers, $3 billion and took 10 years. Today, I can take your genomes, and for reagent cost, it will cost $3,000 and it will take 10 days. This is a remarkable truncation in time. Now, where has this kind of massive expansion have taken place in our history? It's happened before. It's happened in electronics, in computer, and intellect. Remember, there were cathode tubes. Many of you in this audience don't even know what a cathode tube is. But you had then transistors, with itty-bitty transistors radios, which when I was growing up, it was really remarkable that you could carry a radio in your hands. That gave us integrated circuits, it gave us computers and improved devices all the way to your little cell phone. And there was value from hardware. You know, from RCA to IBM, they were the kings in that era. But subsequently, from the late 1970s on, another event happened. It has nothing to do with the hardware. It has everything to do with the connectivity that the hardware allowed, the speed, the volume, the capabilities that gave us the internet, voice over IP, the e-commerce. And this is all because of value from software, connectivity, and data access. Now, if you had asked anybody in 1969 that there would be a whole new industry that is based on not soft sale of software, but your interactivity on something called the web, people would have said, you're crazy. And if you had said that the valuation of Google will exceed the valuation of IBM, software that had nothing tangible in your hands, as opposed to a piece of equipment, people would have said, you're crazy. but this is what actually happens. Something that was created that you never anticipated from this increase in capacity, reduction in cost, and this voluminous ability to connect. Well, this is actually what's happening in genetics. Remember the Moore's law, the idea that the cost will go down, speed will go up, and by twofold every 2 years. And that was projected all the way to, and it turns out to be true. This was actually what the Moore's law had predicted, and as I mentioned to you, the data suggest for transistors all the way to the motherboard, that was what's happening. But this green line is what we're seeing in sequencing capability. It's actually exceeded Moore's law. So consequently, what you have seen in that transition from hardware to software to the internet is going to happen in genomics. We just don't know what it's going to project to be. It's going to happen even faster than what you had experienced with the internet. Now, for genetics and genomics, what we're doing right now is mutation detection. We sequence, we say, well, you have this mutation or that mutation. The genome analysis, then we move into whole genome sequencing, not just one gene, but your whole genome at one time. And what we're doing right now is iteratively improving the sequencers so they're faster, more powerful, and cheaper. That's value from hardware. Very soon already we are talking about value from software and the connectivity, massive biocomputing, genomic medicine, synthetic biology, genetic security. The idea that all your genome and everything that you know about yourself and health can be stored in some kind of cloud that you can extract whenever new information comes about that can inform you for better health. Can you imagine that that's kind of completely reversing the ownership pattern of your medical information that used to be locked up in files in a single hospital, now is in some kind of cloud and a web? Now, this inevitably has an impact on society, and I would argue on the whole is for the better, but let's talk about some of that. Genomics has given us speed, comprehensiveness, I mean everything you can read, precision, everything you can read precisely, and reduction in cost. Your sequence for, quite frankly, less than the cost of a spring break. And that you can keep forever because it ain't going to change very much, hopefully, in your lifetime. Now, I'll give you some examples in cancer. This is a disease called chronic myelogenous leukemia. It's a disease that we know is involved with a single genetic abnormality characterized by this chromosome spread. It is a translocation, a swap of genetic material between chromosome 9 and chromosome 22. In 1960, that was when it was found. It was called Philadelphia chromosome because it was discovered in Philadelphia by well-known cytogeneticists. But it took many years with cloning capabilities for us to realize that the breakpoint between this abnormal process, the generation of a Frankenstein gene, actually occurred very precisely between a gene on chromosome 22 called BCR and a gene on chromosome 9 called ABL. This actually breaks the BCR, breaks the ABL, join them together, and in that process, it activates this kinase, this enzyme called ABL, into a oncogene, a gene that, by virtue of being on, it drives cancer production. This signature, that's BCR-ABL, is only seen in this one leukemia, not in any other diseases, and so therefore it was a poster child of a genetic mutation in a cancer, not one that you pass on to your children, but the one that was a mistake in one of the replications of your cells that caused actually a cancer. Now you fast forward a little while and you find that ABL, being a kinase, being an enzyme, is amenable to be blocked by some kind of inhibitors, and chemists have gotten very, very good, so good that they had structured a small molecule that will fit in the enzymatic pocket of the ABL kinase to block this oncogenic function, the function that causes cancer. And this was actually prepared as a drug called Gleevec. And by the introduction of this one pill taken once a day, This is actually national survival data. This is not a clinical trial. This is the nation's experience in CML. A disease that if you had it within an average of 3.5 years, you would be dead of the disease, is now the mortality fell by 50% in 5 years, is now even down lower. A death sentence disease is now a chronic disorder. It's not good to have, but at least it can be your life can be extended significantly. Now, this, however, is the timeline from 1960 to 1990 to year 2000. It took about 40 years from the discovery of this molecular abnormality in cancer to actually a treatment, effective treatment of the disease. 40 years, one full generation time. Fast forward this in 2007, when a Japanese group using genomic technologies, both sequencing and molecular approaches, analyzed one lung cancer patient from a 62-year-old smoker and found a breakpoint, very much like the BCR-ABL, where ALK is another kinase, another enzyme, that is broken into by another gene called EML, and this actually induces cancer. It's been proven by generating this variant, this mutant, and injecting it into the germline of mice and actually causing cancers in mice. Because it is the same kind of enzyme, the chemists already had some compounds on the shelf and found there was one that actually fit the pocket extraordinarily well. That was quickly developed, and now there's crizotinib, which is that small molecule that will give a 60% response rate, but only to that 6% of patients with lung cancer with that mutation. Now, you might say 6% isn't a whole lot, but if you were one of those 6% individuals, you want this drug, because there are not too many other good drugs for lung cancer today. But from the beginning, from the publication of that paper to actually the passage of, by the FDA, the approval of the FDA for this to be used clinically was 4 years. 40 years, 4 years. This is really dramatic. Dramatic to a point is that you no longer have to say, I might die of this cancer, but I hope my children will survive if they have it. Now you even have a potential hope to say, boy, if I live long enough, they will come up with a solution. This is really quite remarkable. Now, moving forward to this is that genomics, recall, is the ability to scan entire genomes, to record it, to be able to analyze it. So to sequence many genomes to understand essential genes is exactly what has been happening. And to do so, we have actually constructed a parts list of an inventory of many genes and many disorders. And in fact, one of those is the assemblage by the Craig Venter group of taking bits and pieces of a genome of a particular mycoplasma and then reassembling them into a new organism. This was called a biological genetic reboot because it was actually pressing a button that fragmented the DNAs and allowed it to come back together. This is a proof of concept of what we now call systems synthetic biology, the ability to synthesize entire organisms potentially from scratch. Now, we're not there yet in creating whole organisms, but we've been able to exploit that in a significant way. Jay Kiesling at Berkeley was really attacking a very important problem, and that's malaria. And we're funded by Bill and Melinda Gates Foundation. Malaria, as you know, is a scourge of mankind, has been with us for tens of thousands of years and has molded our genome. And in fact, right now, you used to be able to treat malaria with quinine, then it was chloroquine, but they're all now resistant. And the only drug that works is a drug that was extracted from a Chinese plant called Artemisia, the Artemisia plant here. And the drug that's extracted is a natural product called artemisinin. And here's an example of treatment of severe malaria with a drug that used to be used, and it doesn't work anymore, to artemisinin that's here. Now the problem is that artemisinin, by virtue of this of natural products, is actually expensive to extract, about $10 per treatment. But if you think about it, this is actually, we can afford it. But in many of the malaria-infested countries where they spend $10 per person per year per capita for health, that's not possible. So what their goal was to use the biological knowledge to create artemisinin at a very cheap rate. Now you could say, well, you're a chemist, you should be able to sequence it, but many of these natural products are, for those of you who are chemists in the audience, you know, are not straightforward. There's stereoisomers, the different formations, difficult, especially difficult to construct, to synthesize in bulk. And so the only way to do it is try to recapitulate the biology of this plant in some other vehicle. And that vehicle is baker's yeast. What they did was actually take all the components, the enzymatic components that create artemisinin by careful study of the plant, reassembled them in the yeast. And it wasn't that straightforward because once certain exogenous genes were introduced, it has a spillover effect that caused toxicity to the yeast, and some genes had to be knocked out in order for it to survive. So a lot of crafting took place, and finally they were able to create a yeast strain with this exogenous DNA, outside DNA transfected in, built within the yeast, the entire biofactory to synthesize artemisinin. And what he was able to do was actually bring it down from $10 a day to 10 cents per treatment, which at this point is potentially financially doable in some of these countries. And this is a very important point. Now the same thing is happening in biofuels. You heard that people use switchgrass and engineered switchgrass to actually have the biomass that can be converted to biofuels. But another very interesting approach, an approach that when I was in Singapore, one of my scientists were really working on, was taking algae and actually engineer it in such a way that it produce fat droplets inside its cell. And these fat droplets can be actually harvested, and biodiesel can be created from it. And right now there's a number of both public laboratories as well as private companies established to try to find ways to create a robust generation of biodiesel and algae. Recall also that algae uses photosynthesis and in fact is a carbon sump. So in effect you have a process whereby you can take carbon from the atmosphere, convert it into biodiesel, and it's a cycle that can be used over and over again. Now, the very interesting thing about any technology is that it not only changes your realities, but it also changes how we think of ourselves. This is where structure dictates content in many ways. And what do I mean by that? The fact that you have a genetic code that makes you, every single part of you is dictated by that genetic code in you. 3 billion base pairs give you who you are in your complexity. So your genetic self has actually boundaries. It's not boundless, which means that it can be completely discoverable by some kind of process, and in this case, by sequence. Now, do I really think that will actually happen? The answer is probably not. It's way too complex to do that. But the finiteness of that 3 billion base pairs and the ability to put that in a string of order suggests that someday that is actually feasible. So where are some of those, those scientific thought processes that had a huge impact on culturally and conceptually on us as human beings? When the first time we accepted that the sun didn't revolve around the earth, that we are heliocentric, that the sun was where we circled around. Remember the uproar that caused. We're no longer the center of the world. We're no longer exceptional. The germ theory moved miasma, you smell bad things and you're going to get disease, to a finite vector for disorder that is extractable, growable, and treatable. So for the very first time, we had something to hook onto that infectious diseases could be defined by man and not by God, and also treatable. And then the final aspect is, remember, we used to think the universe was always infinite, never began, it will never end. But the Big Bang Theory, which by far right now, as far as I know, there has been no refutation of that, suggested there was a singularity about 14 billion years ago, that everything began from this one Big Bang. The concept that the world, the cosmos began in a singularity that exploded is mind-boggling. And it changes yet again our concept of ourselves, not as the center of the world, but as a little speck of life in a wide cosmos. Now, where genetics is so important in how we think about ourselves and how we govern ourselves and how we actually hurt each other through history is genetics and racism. At one point, we always thought that certain ethnic groups were inferior, genetically inferior to us, closer to animals than to human beings. There was a theory that was very prevalent that human beings didn't start from one branch necessarily, but started as individual branches, individual shoots coming out simultaneously in many different continents. This was primarily with the disbelief that a inferior race, whether it's Asians or Africans, could ever be our brethren. Well, this really changed as we learned more and more about genetics. And the nail on the coffin came with this paper by Alan Wilson and Rebecca Kahn in 1987. Using molecular techniques, they tracked the origins of mitochondria. Now, let me step back for a second. The Y chromosome is only in males. It never recombines, and therefore, your Y chromosome is the history of all your male ancestors from the dawn of time. Your mitochondria are from your mother, okay? There are exceptions, there are diseases even that have heteroplasty, but your mitochondria on the whole are from your mother, and therefore, by sequencing your mitochondrion, you have a history of you and your mother to the beginning of time. So what they did was using mutational markers and tracking and using mathematical models came up with, to them, an irrefutable observation that we all came from a mother in Africa, every single one of us. Since then, this has proven to be absolutely, completely true. There has been no incidence where this model has been broken. Now, what does that mean? It says that all of us originated in Africa, and by migration we went to Asia. From Asia we crossed over to the Americas. We went up the Levant, which is the Tigris and Euphrates and so forth, into Europe. And in fact, this is now called the African Eve concept. And what we have here is that Africans are actually the root of humanity, and Caucasians are a little bit of a tail of humanity. In fact, the least diverse ethnic group are actually Caucasians because they are of one branch of humanity that actually broke off very early in the game. Now, this actually talks about eugenics, and most of you know what that means. Eugenics is the act of trying to improve the genes by culling the herd or by shaping the genome in one way. Of course, you can't shape the genome very well in humans, but you can cull the herd. And the issue here is, and remember, there were legislative approaches in this country to sterilize what was considered genetically inferior people. And by the way, it's not because America is evil. It was at a time when virtually every elite class thought that the only way to prevent your culture from falling into genetic degeneracy was by culling the herd. Now, what's wrong with this concept, though? What's wrong with culling the herd, as it were? Because that culling the herd framework was really came out of animal husbandry and with horticulture, okay? The horticulturalist view of the world is you breed the most beautiful plant, beautiful to you, but you're not breeding the strongest plant. What is beautiful? Something pleasing to the eye that only lasts for a short period of time, or something really robust that won't go away? It's pretty ugly, but will survive. Which is the best? Now, nature will tell you robustness is the only thing that matters. But the only reason why we believe that something is beautiful should survive is because it pleases us. It has nothing to do with survivability, and therefore, this concept of an ethnic group being dumber than we are may not be the most important thing, because perhaps the most important is your emotional quotient, your ability to hunt in packs, your ability to withstand infection. Is that not equally important in robustness in humanity? So robustness is really context specific. There's really no universal form of genetic goodness, and I think that is actually the reality. In truth, you can put behavior in the genes. All you need to do is actually look at your pet dogs. A Cocker Spaniel behaves differently from an Australian sheepdog, differently from your Westie, different from your Doberman Pinscher. And in fact, the dog breeders can give you a catalog of average behaviors of these species. Now, if you can breed behaviors in dogs with only 400 to 500 year history of breeding, quite frankly, you can do that to human beings. But the question is, what is good? I think that's the bigger question, because robustness is context specific. Robust marine animals do very poorly in land. What's robust? It's relative. Big dogs have shorter lifespan than short dogs. What's robust? A big dog that can chew up the little dog, or a big dog that's going to die faster by twofold than your little dog. A lack of hair gave a human advantage for sweating and heat exchange, hunting as a bipedal pack, but you're pretty bad in cold weather compared to animals with fur. Robust individuals versus robust populations. You have a moose that is very robust but lives alone versus a pack of wolves that by themselves are not going to survive. Which is more robust? It's like an individual bacterium for microbes in a biofilm. So in many ways, genetic diversity in populations is beneficial very much more than monocultures. So the concept of the master race is a fallacy. It's like the master potato that caused the potato famine in Ireland, where the Incas already knew that when you plotted potatoes, you need to have different varieties plotted at the same time, because you never knew when a blight would actually come through. Now, there are many good things about genetics. I've told you about the physical, the technical advances that it's given us to treat cancer, but there are many more examples. And also changed our view of the world, I believe, for the better. But there are some caveats to this, some concerns that I have. First of all, this is a very interesting paper in 2005 that came out of work 5 years before that. In 1918, there was a worldwide pandemic of influenza. It was one of the most virulent strains that were around. It killed at least 20 million people worldwide. And killed many young individuals. It was devastating. But somehow, as in many of these devastating epidemics, it went away. Now, what has happened is that we've always had flu, but this strain was really a bad one. So what caused it to be bad? So what happened was a group of scientists went and dug up some pathology slides, some samples from a person who died in 1918, the lung samples, okay, because influenza affected the lungs, scraped it by polymerase chain reaction, or PCR, from the knowledge that we have of the general flu sequence, PCR of each fragment, put it in a catalog, then pieced it together into a real virus. And then what they did was tested it in test animals. Now this is where laboratory flu, its effect on survival, which is 100% here over time. Inject that into an animal, and that's not going to cause them any problems. But you inject this reconstructed flu virus, and all of a sudden, all of them died. And with a slight manipulation of the genome, it was even more virulent. So those who are involved in germ warfare talk about sequence, to reconstruct it, and to weaponize. This is the normal. You sequenced and reconstructed it, and then you weaponized it by making it even worse. It is a hop, a skip, and a jump for you, if you set up a laboratory, to reconstruct a virulent virus if you really wanted to. In fact, remember the Anthrax scare that came out, that somebody sent both the members of Congress and the president this white powder that turned out to be anthrax. Well, this was a weaponized anthrax that they tracked using DNA sequence back to the laboratory where it came from, because there was a signature embedded in that and registered to where it came from that gave us this. So this is one thing that you're all going to have to think about as citizens, of how we're going to govern this, how we're going to manage it, and how we will control it. Now, who is this person? Say? Right, Henrietta Lacks. This is actually a very, it's raising a very important issue for us. Henrietta Lacks was an African-American woman who had cervical cancer. And unbeknownst to her, she was never asked, a piece of that tumor was taken out and a cell line was created. That cell line was a absolutely profoundly important cell line, because prior to that, there was no reproducible, immortal human cell in culture that could be used for cell biology experiments. They would do some primary tissues, but they will all die. And you couldn't share it because they couldn't be propagated. And one of the reasons why microbiology was so important, really advanced, was the ability to grow the bacterium on plates. So then you can share with other scientists to actually reproduce. Well, that was bad enough, but again, in all fairness, that was a standard in the day. Is certainly and gratefully not the case. Well, of course, there was a book, a very important book, I think, was written, of The Immortal Life of Henrietta Lacks. And of course, it raised a question of what right did she have, what right did the doctors have in taking this? And by the way, this cell line became this substrate for how all of, much of pharma actually created things like vaccines and tested many of their drugs. So the benefit that you have from Henrietta's cells is really immeasurable. But then something else happened. Remember, your DNA is a record not only of you, but of all your ancestors in one way or the other, and a projection of what your children will look like. And with the sequencing technologies, several groups sequenced in high detail the genome of Henrietta Lacks and then put it into the public domain. The problem is that they didn't ask the family. Now, Henrietta Lacks is dead. And by most convention, even today, if a person is passed away, the work with their cells does not require informed consent from the patient, as long as there are certain security issues are there. But the problem is everybody knows this is HeLa cell. Everybody knows from that book is Henrietta Lacks, and their family is around. And this actually has information that is pertinent to every family member, every descendant of Henrietta Lacks. Well, what happened was that, gratefully, I think we've matured as a society. And in that process, there was a agreement struck with the family that the family would actually have a say in how the sequence could be distributed to other people. Rather than to be completely closed, which won't help anybody, they just wanted to make sure that, that their family secrets are actually kept are given only to people who are responsible. This is part of the governance of important technologies that you will have to deal with. Now, let's fast forward. Where might we be? What happens when our sequencing costs come down to $100 per genome? And you can sequence almost anything within a flash. In which case, we can sequence the genetic components of all living creatures. All 10 million creatures, perhaps. With this evolutionary part list, we can actually reconstruct biology, biochemical cells, organisms, even the concept of reconstructing an extinct animal. Jurassic Park is a bit of a stretch, but it's not that far of a stretch given the possibilities that we have. One of the key issues is to master complex systems. And when we say we harness whole microbial communities, and we can completely monitor the life fluxes on Earth as if we were having a EKG on Earth on a real-time basis. Where might we be with this kind of genomic information? Recall that at one point all your medical records were in a hospital on paper. You went there, signed an informed consent, carried it somewhere to somebody else. And in fact, long ago, hospitals considered your information as property of the hospital, not of you. Now that has changed. But what's happening now is that perhaps your medical information, including your genome, by the way, with all the secrets that are there, could be yours locked up in a secure file in a cloud somewhere. And that two things might happen. One, you will release it only when you think it's appropriate, like giving access to only the right persons that you want. The second is that you may want to have a steward, an agent. You hire that person because you have so much information, you don't know what to deal with, how to deal with it. But that agent is going to interrogate your data, match it against new knowledge that's being generated on a fast clip. So that anything that is new, they're going to buzz you and say, we found something about Alzheimer's disease. We don't know whether or not it's important in your genome, but would you like to know? The answer is yes. Okay. This is actually the consequences of knowing. Do you still want to know? The answer is yes. This is, do you want to know whether you have these abnormalities? The answer is yes. And then you say, who do you else want to know? Nobody else. So you have that. Because here is the thing. Your genome isn't about to change. But you can imagine this kind of information inversion whereby in the past, you were the store of all the medical information. You went to the doctor, a blood test was taken, a blood test is taken again, a blood test is taken a third time. But here, you get your genome sequence, and you don't need to sequence it again. The issue here is it can be locked up, and the information can be extracted whenever it's actually needed. Now, there's also, think about potentially the social applications of personal genomics. What's to stop you from asking your potential spouse, can I check your genome? No, think about it. Some people say, can I see your HIV status? Why not, can I see your genome? It will probably happen, as not because it's enforced by anybody, it's because you, as a member of a free society, decide that's important to you. Does that mean there's going to be greater truthfulness? Now, the computer forensics, I believe, I'm just watching this, has really caused a lot of people who would be chronic liars to be truthful because they know they can't get away with it at all. Perhaps our genomic forensics will give us actually that same. Now, the issue of expression of personal freedom is already happening. The Personal Genome Project is over 1,000, actually is more like 5,000 people who volunteer not only to get their genome sequenced, but will deposit all their medical information and their names on the web for everybody to see. Why? Well, why do you have Facebook? Why do you tell the world that you're dating somebody? So these are the personal choices that are coming out that have really profound ramifications. And 23andMe, you've heard the commercials, right? Okay, it tells you not only maybe some health issues, but it tells you what your genealogy might be. Oh my gosh, I'm part Viking, which was, But then what's happening also is that it's a chat room, because there's actually options for you to say, I'd like to meet all people who are partial Vikings and have a conversation about our mutual ancestor. Now you can imagine that that's going to extend to, I kind of like Vikings, and maybe I should get married to a Viking and actually ask for a person with a Viking, a genotype. What's to stop you from that? People have married for worse, and with this. And this was all culminated in a great movie. How many have you seen Gattaca? It's great, isn't it? I mean, it's really beautiful acting. It's genomics and reproductive choice, okay? Now you can think this is crazy, but think again. In the Middle East, in the Gulf states, consanguinity, do you know what consanguinity is? It's marrying related individuals. 60% of marriages are to first cousins or to niece uncles. Okay, 60%. 60%. Anywhere from 40 to 60%. The rate of recessive disorders is relatively high, and it becomes a burden. It's a health burden for the society and for the family. There is some discussion amongst only the more liberal parts of the Gulf states that perhaps when first cousins marry, they should have their genome checked for clear recessive disorders. Why not? You can do it now, and you can actually have a way to actually prevent some of these recessive disorders. In fact, one of my colleagues who's a geneticist in the Middle East basically said, it's probably easier to actually have this genomic pairing than it is to change a culture that's been around for 1,500 years. So finally, this is just a personal statement. There are any new technologies, there's going to be advantages and concerns. They truly are ethically neutral. It's really their applications that determine goodness and evil, and the failure of human judgment is a problem. Now, I'm not one to say, guns aren't bad, it's people that are bad, and we should let all guns, any kind of gun, access to any kind of guns. That's your decision and what have you. But I think you have a right to carry a gun, but it has to be monitored and marshaled and stewarded in the right way, just as much as you have to get a driver's license to drive a car. Society has decided that you need a driver's license to protect not only yourself, but everybody else. At some point, we're going to need to think about how we're going to do this with our genomic information. Now, just in case you think that any of this is totally, has been around for a long time, I want to show you this one, which is really astounding. Did you know that in the history of our species, there has never, ever been the explosion of culture, society, and population than we have in the last 250 years? In other words, all this stuff that we're seeing now, from the science to the social issues to some of these massive ethnic issues, is really very new, even though we as a species have had 120,000 years of, at least 120,000 years of history. So let's not berate ourselves. This is new, but we're pretty smart. The key is for people like you, the people who will be leaders in tomorrow's world, to be intelligent about what the technology brings and to be sufficiently principled so that you can devise governing patterns and monitoring processes that will not only allow progress to proceed, but to do it in a safe and effective manner. So I want to thank you very much, and I'm open to questions, so thank you.